Coronavirus is a conspiracy by Jack Dorsey and YouTube to get more subscribers and ad revenue. Dun, 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 dun.
I mean, you've heard all the crazy crap, I'm sure, about Bill Gates is like gonna profit off the vaccine and stuff like that. This is so he can put microchips in us and all that.
Yeah, the vaccines are going to contain microchips now because we need those for vaccines. So clearly, hilarious.
Yeah, and yet everyone carries around their phone and no one thinks about that, listening to you or anything like that. But oh well, that's another conversation.
Yeah, it is pretty funny. Shoot, I just closed my chat window. Why did I do that? Social media, live stream, restream chat. All right. We should be live now. And so, people, is the sound working? The six people in the chat right now. All right, and then give that like 15 seconds because of the lag delay to actually hear stuff. TJump, that is correct. TEA jump. Yes. Gad Sad is my dad. That's his name. That's fantastic.
Well, that's interesting. I follow him on Twitter. I've not read any of his books. I have I have not read a lot of evolutionary psychology literature. I'm skeptical of it, but I'm curious to read it just to kind of see what they have to say. And so,
why is that?
Unfortunately, I haven't.
Skeptical of Evo Psych?
Yes. Unfortunately, a lot of it is sort of co-opted for agendas, more or less. And when you listen to them make these explanations of, oh, well, this is why we do this, and you listen to it like, wait, what? No, that doesn't make any sense at all. So obviously, some of it has to be rooted. Some of our behaviors have to be rooted in genetics. Religion is probably, I would say, almost certainly a genetic phenomenon in some capacity. But do we know what the origin of religion was? Did it serve us some natural selective benefit? Was it just a meme that caught on? Which is the Dawkins explanation. Well, we don't really know because we don't really have any way of testing that. And we can come up with all sorts of hypotheses to explain it. But at the end of the day, we don't really have, unless we have like Australopithecines to sort of poke and ask them, hey, do you believe in God? We can't really go very far with that question.
Oh yeah, I totally agree on that front. That Evo Psych is pretty much hypothetical because we can't go back and actually see how the psychology of the things were affected by the environment in order to produce something at the time period. So it's all pretty much just guessing, and that's the best we can do. But yeah, I definitely think that there's some merit to it. Like you said, religion, I think definitely there's some evolutionary basis, morality, and I think there is a lot of work we can do in neurology, though. Like we can see the correlates between like conservatism and certain neurological patterns, or liberalism and certain neurological patterns, and then from that we can probably infer the Evo Psych, as long as we have that kind of hard data today that we can base it off of. So that realm of Evo Psych, I think, is pretty substantive in that we can make predictions about how it infected us in the past and how it can be seen in today's neurology. That's beneficial. But just projecting back into 10,000 years ago, say, I think this is how it happened, is not very useful.
Right. Yeah. Yeah, absolutely.
Awesome. So, Jackson, thanks for coming on. I really appreciate it. Would you mind telling us a little bit about yourself and your YouTube channel before we get started?
Oh, sure. I'm just a biology student. My YouTube channel focuses on biology, teaching concepts in evolutionary biology, as well as occasionally combating creationism, though I don't do that as much as other people who sort of focus their whole channel on that. I try more to focus on sort of the positive teaching of different topics. But I do debates also occasionally. My most recent video is a debate I had with Dr. Fazal Rana on universal common ancestry.
Yep. I love his nickname. He likes people to call him Fuzz. And I'm like, that's what I call my dogs, Fuzz. So the reason I invited you on is because I wanted to talk about information. I think it's one of the best arguments that theists have, because if we say we look at DNA as a code, all codes are made by a mind as far as we can tell. Therefore, DNA is made by a mind. That is a valid argument. That makes sense. Obviously, it wouldn't apply today because we have lots and lots of testable predictions that have been confirmed by the natural hypothesis. But if we imagine that we lived back in the discovery of DNA, like the day it was discovered, like, oh, this looks a lot like a code. all code seem to be made like a mind, that seems to make like a mind, that probably would have been a reasonable argument back then. Because if we see like this inorganic matter forming into some pattern that is very code-like, and then to see that code produce something completely different, that's a very strange phenomenon. And that's the phenomenon that theists keep describing with this information thing that most of them have no idea what they're talking about. But it is a real surprising phenomenon that we can see inorganic matter form into something and have that something build a completely different thing that seems to have no relation to the original thing that it was built by. So, could you tell us a little bit about this information thing that is in matter, and how does it do that?
Well, there are my first problem is with the whole concept of like information is there are mathematical concepts of information. There's a whole information theory, and there are formulas surrounding that. But when we're talking to theists, especially young earth creationists, who are usually the ones in the debate circuit having these conversations, information doesn't really mean anything at all. DNA has information in it, but obviously DNA is a molecule. It is a physical thing. So information, in a sense, must be a physical thing, right? It's some section of this DNA molecule which has some effect. Well, and so when I had this conversation with creationists, and RJ and I wrote a whole section about it in The Rocks Are There, is, well, what do we mean by information? What is the definition? And most of the time, creationists will either give it a definition which is completely nebulous, or they'll define it as something which is not useful in any sense. We talked about how Mike Riddle was part of a whole group of creationists, and their definition of information was basically like words that tell you to do something, which is like, okay, well, that doesn't apply to DNA at all. So why are you using this term? And you're right. We have now, or we have experiments and observations and inferences about how DNA has changed throughout the history of life. I think the either the field observations or experiments are the most powerful, especially when talking with creationists, because you can kind of show them, okay, here we have this, we directly witnessed this. Here's how it was before. Here's how it was afterwards, or is afterwards, and we can explain this purely with natural phenomena. We don't have to invoke creator, what have you. And so I actually I brought one paper with me. Let's see, there's a chat right here, so I'll just send you the link right here in our little side chat.
Oh, cool.
So this paper is about a species of crawfish, Procambarus phalax, who in the 1990s, one of them underwent a whole genome duplication. So it went from diploid, having two sets of chromosomes, to being triploid, has 3 sets of chromosomes. And as a result of this, it became parthenogenetic. So the virgin birth, it doesn't have to mate with a male, because they're all female, to have offspring. They clone themselves, basically. And so they also have slight morphological differences in comparison to the parental species, such as they have slightly larger clutch sizes, and P. virginalis is slightly larger on average. So we have genetic differences and morphological differences as a result of a whole genome duplication, which is a natural event. And so you have sort of everything kind of this really good, tight argument against the idea that changing code needs a code changer, right?
Definitely, absolutely. But the reason I think this is a really fascinating argument is obviously the way the theists phrase it usually leads down this path that you're describing, is that they have no idea what they're talking about. They have these ambiguous definitions of information. But I want to try to steelman their argument, try to make it into something the best I can. So I'm going to play the theist here. And I'm going to say, so the way I'm defining information is that we see inorganic matter forming some kind of a form. And that form is like a factory that it builds something else. And this new thing that it builds seems to have no relation to the original thing itself or the way that the original factory was built. Like, say, RNA forms on clay, or RNA then builds DNA builds proteins. Well, it doesn't seem like there's any kind of necessary relation between the proteins and the RNA. Like, how did RNA get from that stage to then building proteins? Because it seems like the only way it could do that would be some arbitrary rule set was imposed on the RNA to make it build the proteins. So, because it seems like it could build anything, it could build lots of different things. Why would it choose to build a protein, and how would it choose to make that particular protein as opposed to just a random jumbled mess of nonsense? Because it seems like this RNA is like a factory, like a Tesla car factory, and it builds a car, but the car has nothing to do with the factory. Like there's no car imprint somewhere in the factory that makes it build a car. You could just change some of the code and it could build something else. And RNA seems to be the same way. So where is that coding that makes the RNA the way it is, that so it can produce the protein as opposed to just anything else?
Well, my issue, sort of when I have this area of conversation, is this kind of what RJ likes to call the origins or bust. We have to go straight to abiogenesis. And I must confess, I'm not a biochem person. I'm not a cell person or a protein person. I'm an organismal person. My general understanding of this is that you had RNA first. RNA catalyzes its own synthesis, and you have the ribosome, which is involved in translating messenger RNA into proteins. The core of it is RNA, right? It's the ribosome, and so RNA was doing these things on its own. It was replicating itself and making proteins, on its own. And then you have DNA, which came later because DNA requires a number of enzymes like topoisomerase and polymerase and all this for its functioning. And so, what is there a particular reason that it produces, or that mRNA is translated to make proteins? I'm not really sure. You'd have to ask somebody who's more in the biochem aspect of that, someone who really studies the origin of life stuff. In evolution, though, typically what you see is things building on other things. So to go back to the factory analogy, it's not really like you produce a car, you're producing airplanes, and then one day, the factory just makes a car. It's more like you're producing cars. Well, now you're producing a slightly different type of car. You already had sort of the base thing, and now you're sort of adding on to it. And that's, there are things like acceptation, where you take something which had one purpose, but now you're crafting it for a totally different purpose. But you're using the same genes, right? So like the fish fin and our limbs are crafted from the same genes, or the developmental genes which are involved in that are exactly the same, like Sonic Hedgehog and things like that. And so it's not really so much that it's a completely different thing. It's you're slightly altering what was already there. And I feel that that's
so
also...
what I mean by completely different is I mean that the RNA and the protein are different, not that the different proteins are different.
Sure, yeah, they're different molecules, yeah.
So that's the thing that I think most theists are trying to get at when they're making this argument, is that how do you go from, and we can ignore abiogenesis and just go with living cells right now, that you go from something like in a cell, you go from RNA to DNA to proteins. Like, how does the RNA make a... What?
DNA to RNA to proteins.
Yeah, okay. Well, because the RNA world hypothesis, So DNA to RNA to proteins. How does the DNA know to make RNA? Like, is that... Because it seems like that's a rather arbitrary thing, that DNA could make anything. Why is it making this completely separate thing as opposed to another one? Because how did it get that programming in the DNA to make the RNA as opposed to just a similar DNA? Why doesn't it just like duplicate itself like a crystal? Why does it make some completely new kind of a thing?
Well, that's still an origin of life question. This is like pre-last universal common ancestor, because by the time you get the last universal common ancestor, you have, well, in that instance, you had an RNA genome already, which was translating proteins, right? And so this is occurring far afield of that. So this is, way before the last universal common ancestor. So I would still consider that sort of an origin of life issue. Why is it DNA is what specifies or is what contains the genes, which are then transcribed into RNA, which are then translated into proteins. That's all very, very deep in the origin of life. That's way at the start. And again, that's not my area of expertise. I haven't really done a whole lot of reading on that. There's probably a lot of really good literature out there on the subject.
Maybe a better way to phrase it would be how. How does DNA contain the information that can produce a completely separate molecule?
So your genes are so when you have a gene, which is a segment of DNA, and it's transcribed by RNA polymerase to produce this little segment of messenger RNA. This messenger RNA, then each of the sets of three, it contains sets of 3 nucleotides, which are called codons. Each codon, each little set of 3 nucleotides, is then read by the ribosome using transfer RNA to produce a single amino acid, right? So these 3 nucleotides are mapped onto 1 amino acid. And so it reads the first little 3, you have an amino acid, then moves over, you read the second one, it has another amino acid. These 2 are joined by a peptide bond, and then moves over to the next one, another amino acid, peptide bond, and that's how you generate an amino acid sequence, which you then fold into a protein. That's the how of how that happens in like a modern cell. But I believe there's some argument that the original codon system would have actually been 2 nucleotides rather than 3, and that's probably why there's variation among different organisms with regard to why certain codons can specify amino acids, but why you can change that codon and get a different amino acid, things like that.
So essentially, it's the information is contained in the physical shape of the DNA. So the physical shape forces it to bond with certain molecules in the vicinity that forces it to build a specific kind of bond. Kind of like if you have magnets, you have a positive and negative, and the positive always goes to the positive, the negative always goes to the negative, which causes it to form a particular shape. And that particular shape is, of course, the thing it's producing. So the DNA can produce something because of the way the physical shape bonds with other physical shapes in the vicinity. And that's the information. So the information is just the physical shape. So it's not like a code that's been written somewhere in the brain of the DNA. It's literally just the physical shape of the DNA that contains the information.
Yeah, I mean, if you combine what is it, hydrochloric acid and sodium hydroxide, there's nothing inherent to either of these molecules which says cause an exothermic reaction. It just happens because when you combine them, they're going to switch. Their molecules are going to do this whole reaction, and the exothermic reaction is a product of that, right? It's just the way that the properties that they have that are inherent to these molecules. I would say you could call the codon to amino acid the genetic code, which is fine. It's everyone calls it that. But would I say DNA itself is a code? No. And I have a set of reasons as to why.
Yeah, that's an interesting question because it is referred to as a code in lots of papers, like academic papers from scientists, that DNA is a code. So why would you disagree and not call it a code?
Well, like I said, there is a code aspect. The codon to amino acid, I would say, is a code, is a genetic code. But DNA contains a lot of elements which don't do anything, right? And so if you take something like a pseudogene, like gulo, which can't even be transcribed, I wouldn't say that's a code, right? Would you say it's a code if it's something that can't be transcribed at all? It participates not at all in the functioning of the organism.
I think I'd still probably define it as a code just because of the patterned way of the ATCG thing. So I think it would probably be a code. It'd just be a bad code. So I think I've written tons of bad codes. I think there's still code. They're just bad code. So I'd say. I mean. So the way I see it is I think that just because it follows that pattern of ATCG, which is like a system of aligned order of some kind, I would still say it's a code. It's just not a good one in that case, or a useful one.
Well, I mean, I guess if you want to define code that way, then okay, sure, you could consider it a code, I guess.
But so you would not define it as a code because you think it has to be useful in some way for it to be to be coding?
yeah, if it's not regulatory, if it's not regulating either the functioning of other genes or it's not producing proteins itself, then I would say it's not really code, especially since some of them are just random sequences, right?
Yeah.
And so it's like, if you just hit a bunch of random letters, on your keyboard, I wouldn't say that's really code. I mean, I guess you could say it's bad code, which you could, but I wouldn't really define it that way. But that's just me.
Yeah, so what would you take about, like, for example, the encode argument that there's been many additional functions that has been found to DNA. So even though we think that like 80% of it or something was junk DNA, we found out actually there's lots of other functions to it that we've discovered since then. And so that part would actually be code that you just didn't realize was code.
So okay, so the encode argument is so there's a misunderstanding with the encode argument. It's not. There's a difference between something having a function and something functioning, basically. The encode thing measured if there was biochemical functionality in of the genome. Well, the problem is there are lots of things that have biochemical functionality which don't actually do anything, and these are things like spurious transcripts. So if you have a section of DNA and it gets transcribed by accident, it's transcribed well below the threshold of actually working, having a function in the organism, then they would say, "Oh, look, it has a function." Well, not by my definition. I mean, sure, it was transcribed, but it doesn't do anything. Gulo can be accidentally transcribed, theoretically, but I wouldn't say it's functioning because obviously it's not producing a protein, right? And so creationists they point at this thing and they say, "Oh, look, 80% of it's functional." Well, 80% of it may be transcribed, although you could just say, why not say 100% of DNA is functional? After all, at some point it comes into contact with DNA polymerase during replication. So why not all of it, right? But it's not doing anything. It's only about 15% of it is either protein coding or regulatory, right? So it always I get real frustrated whenever I hear the encode argument.
Yeah. So basically, the argument is that lots of parts of the DNA are replicated, but only certain parts of it being replicated actually do anything. And so the fact that it just replicates doesn't mean it's functional. And that's the part that the encode is arguing. Well, if it gets replicated, therefore it's functional, when that's not the case. So because the way most people think about it is, well, DNA would only produce the useful parts of DNA, but like, no, that's not the case. It produces lots of junk and replicates all of it. Only the useful parts do anything. And that's where the confusion is with most creationists think that, well, it's only producing the useful parts and the rest of it doesn't do anything. With like, no, that's not how it works. It's producing all of it and only the useful part does anything. And that's what the difference is between the definitions of function being used in encode versus the classical biological function DNA sense.
Right. Absolutely. Yes. That would be a good way of summarizing it, of the issue.
For sure. But I still think that there's still this huge intuitive problem when you see that there's this just inorganic thing that produces something completely different. And that thing that's completely different is functional. And like you said, like the folding part is really confusing because if it's not folded in the correct way, then it doesn't work. So how does it fold it in the correct way? Because there's tons of different ways you could fold it. Like the folding is hugely complicated. So what causes that folding to be correct? Well,
actually, depending on the protein, it can be folded a number of different ways and still produce functionality. Some proteins are what you call promiscuous, and these promiscuous proteins, you have lots and lots of amino acids involved. And yes, you can fold it in one way and produce a certain function, or if you fold it slightly differently, you get a slightly different function. And so creationists tend to sort of portray this whole thing as, well, either it folds in the correct way and you get your correct thing, or it doesn't and the whole thing just falls apart. Well, no. That's not how you do it at all. You can with minor mutations, you can generate if you have like a point mutation, so you just change one nucleotide. Well, you could generate a different amino acid in this sequence, and so you generate a different amino acid. Well, yes, that could in some proteins that could break the whole thing. Like what's the name of it? The one of the crap, I can't remember. It's a very common disease. It's like you have your sodium channels are don't work correctly, and it's caused by like a single point mutation. Well, anyways, yes, you can have that, but it also could be beneficial if you slightly alter this. You could get a slightly different type of protein, which is beneficial, and that's where the selective pressures come into play.
Sickle cell, is that one?
I don't think it's sickle cell. I can't. No, the sickle cell is red blood cells, but
cystic fibrosis.
Oh, I think that's cystic fibrosis. Yeah, I think you're right.
That was Bird Flu C. William in the chat who got that one right. 1,000 pine points.
Thank you. Yeah. But so yeah, you can have it could break down, or there's actually this process called what is it, alanine scanning, where you replace different amino acids with alanine. And so you can see, is the protein still functional? And actually, in some cases, you can replace a lot of the amino acids with alanine, and it still retains its functionality. So yeah, if that's something you want to look up like afterwards, I'll put it here in our little side chat.
Definitely, absolutely. So, well, yeah, I'll copy it again to the what's it chat again. That's what I've been doing. I've been copying the text and then just pasting it into the YouTube chat, and then people can find it that way. Cool. So but how many different ways are there that a protein could potentially fold? Is it a lot?
That is an interesting question. That kind of comes into like fitness peaks and things like that. How many different paths are there to a particular function?
Well, no. I mean, like, just if we just take the physically possible ways that it could potentially fold, is it like a dozen, 10, 100, 1,000, 10 million? Like, if we just combine all the physically different possible ways anything could fold to produce a functional or non-functional protein, how many different ways are there that could fold?
Oh, well, I mean, if you just keep adding amino acids, it could be like billions or more, I would imagine. You just keep adding them because you're talking about like, well, which is going to form a hydrogen bond with which one? What is this? What functional? What are the chemical properties of this R group over here? You do stuff like that, you're going to get loads and loads and loads of different possibilities, different combinations, things like that. So
absolutely. And so, but not all of them are functional, right? A lot of them would produce a lot of nothingness. And so only a small portion of them are functional. And given that they could bend in all of these different ways, and it seems highly likely that they normally form in the more functional ways, that seems to be an indicator of the fine tuning. Oh, that's such a fun term. So,
well, what I would say to that then is, if you look at things like, or if you look at proteins which are promiscuous and they can bend in lots and lots of different ways, and I mean, yes, by comparison, I'm sure if you look at like, if you have a total morphospace of like protein folding, which is going to be gigantic, but then you only have a small amount of those which are actually realizable, right, or actually functional. And so would I say that's fine tuning? I mean, fine tuning by natural selection, sure, but by any deities, not really.
So how does it do that folding? How does it fold in that particular way out of all the others? What guides the folding process in the cell?
Well, you have different enzymes, things like was it chaperonins? I think. But you have enzymes which cause like hydrogen bonds and things like that to form between different amino acids, depending on and they'll fold in certain ways depending on their function. Like if you have a membrane protein, parts of it are going to be hydrophobic and other parts going to be hydrophilic, and so it's going to be kind of weaved through the membrane. The hydrophobic area is going to be in the middle of the membrane, while the hydrophilic area is going to be on the outside. And so it very much depends on what the function is and what sort of chemical properties it has, and these are all guiding its folding.
Could you explain that in a little more detail? Like, for example, how exactly does it bend right or left? Because the bending is essentially it crumples up into a giant ball, right? It's not like a Jenga thing. It's
Well,
usually
that depends. It depends on the function. Um, like I said, if it's in like a membrane amino acid, it's going to be, sort of going through the membrane. It's not going to be just a ball stuck in the middle of the membrane because it has this. It's going to facilitate the movement of something of materials across the membrane, right? If it's like an aquaporin or something like that, it's going to be a couple of them, and they're all kind of uh, together, like sort of around, and you got water molecules going in and out of this membrane. But again, that's guided by the chemical properties of the protein itself, of the amino acid sequence, which is generating the protein.
so is the protein just generated somewhere in the cell and it just kind of happens to land in the correct place where it does its thing, or does it not matter where it's placed? Is it moved after it's created? How does that work?
Yes, it's shuttled uh, by things like vesicles. So you have okay, so you have your amino acid sequence is produced by a ribosome, right? And the ribosome is adhered to the endoplasmic reticulum, or the rough endoplasmic reticulum. And so then this is shuttled by vesicles to the Golgi apparatus, and the Golgi apparatus can add different little chemicals to it so that this nascent protein can be shuttled to its correct location somewhere in the cell. And again, this is still guided by the thing I think would be important to really get across to like creationists is God's not doing it himself. He's not grabbing it and moving it somewhere. This is all guided by chemical processes, right? It's got a certain set of little markers on it when it's made, and the endoplasmic reticulum uses its little microsomes, which shuttles it to the Golgi apparatus. The Golgi apparatus recognizes it has a certain set of properties, and it's marked it's marked chemically in the Golgi, and then it's shuttled off and it's taken by vesicles to wherever it's supposed to go for its functioning.
Okay, so this seems really complicated. Like, how could it do all these things? So I have this hypothesis. This was done by a god guy, supernatural sky daddy who did this, and you have a hypothesis. This is done by natural processes. How could you show that yours is a better explanation here than the sky daddy?
Well, the I think the fact that this is all done naturally, and as time goes on, we figure out more and more about the sort of evolutionary history of these different organelles. Like all of these endoplasmic reticulum, the Golgi apparatus, these all formed after the origin of eukaryotes, right? The origin of membrane-bound organelles occurred after the endosymbiotic event in which an archaean took in a bacterium, which became mitochondria. And so now with the ability to produce much more cell energy, which is ATP, adenosine triphosphate, you can now make membrane-bound organelles, and you can do lots more reactions than you could previously as a prokaryote because you have a lot less chemical energy to work with. And so the fact that we are now generating evolutionary models for these different systems and for these different organelles, it removes the need for any divine tinkering.
So basically, your model can make testable predictions that can tell us new things that we didn't know yet that the God model can't do, right? Something like that?
Yeah, sure. I mean, if you look at mitochondria and you say, okay, well, this has, a circular genome like bacteria. It has an inner membrane, which is just like that of bacteria. It has its own ribosomes, just like bacteria. You then have to start asking yourself, okay, we don't know everything about how the endosymbiotic event occurred, but what we do have to ask ourselves is, why would God find it necessary to take a bacterium and make it into mitochondria? That seems like this was an event that occurred naturally, and we're just co-opting this pre-existing system. It becomes down to a matter of, well, can we really test the God hypothesis anyway? not really. This seems like a more likely explanation, given that we know all this stuff about it now. So,
Could you give some more examples of the like discoveries that have been made based off of the naturalistic hypothesis of describing these things in the cell? Like we've, if this is natural, then we would predict to see this. We've done this experiment which confirmed it. Those kinds of things.
yeah. So as you all know, Darwin was the guy who, or Darwin and Wallace were the guys
Who's
who
Darwin? I've
proposed.
never heard of this Darwin guy.
No, I know he's this very vague, mystery shrouded character in ancient history, but part of his hypothesis for natural selection was that something being selected has to be functional or useful in some regard at every stage of its development. And so for some things, we can test this, right? We can show that the little hairs, the little fan-like propellers on this species of water strider were useful at every stage. So even if we reduce them, we can still see, okay, the organism survives, and they're just not as good at doing what they were or what they are good at doing with the full complement of structures. And so we can develop models based on, okay, this is how we know this works, and here are these proteins involved, and we can sequence these. And when we look at these, they share homologies with these other proteins. And so it seems to be that there was a duplication event or a set of duplications which expanded this gene family. And so these proteins took on slightly separate functions over evolutionary time or over geologic history. And that's how these structures changed. And so in some cases, like it's ATP synthase, we have more or less the entire evolutionary history of this structure, which is a molecular structure, right? ATP synthase is a type of ATPase, but the function's flipped. Rather than taking ATP and breaking it down into ADP and inorganic phosphate, it combines these, right? And further, the actual structure itself, which does this, is a DNA helicase, which was co-opted for a new function in being an ATPase. And so we can develop these models, which are again testable. We can look at sequence the genes and all that, and look at the proteins. Whereas the God hypothesis is really more, it's done, right?
Definitely.
It's too complicated. Couldn't have happened.
So yeah, but do you know any of those studies offhand that anybody could look up if they wanted to? Like, for example, because the one I like to go with is, we're not usually talking about abiogenesis, is the RNA on clay model. Do you know anything about that? Like, could you explain that to more detail to us?
Not really in much detail. I know the Montmorillonite clay sort of acts like a sort of lipid vesicle, and so you can get organic molecules in there, and they're sort of insulated, and they have a space in which they can safely reproduce. And so theoretically, these could, RNA is imperfect at reproducing, and so you could have mutations which slightly change its functions over time. But that's about as far as I know. I'm not really, as I said, I'm not really super familiar with origin of life stuff.
How do you respond to they're all man-made. They're all the scientists are just fiddling with the data, and it's not really the natural processes doing it, it's the scientists doing it.
Well, the way that I would respond to that is, well, for one thing, we are in a different, the conditions we are living in currently are vastly different from what are proposed for the origin of life. And the origin of life, the atmosphere is largely carbon dioxide. The oceans are more acidic as a result, because carbon dioxide is dissolving in the oceans at a higher rate. These events are occurring on the bottom of the ocean, whereas today, if when you go to these places where the origin of life is hypothesized, like alkaline hydrothermal vents, it's, if organic molecules are being produced at these sites today, odds are whatever organic molecules are being produced are going to be eaten up immediately by either bacteria or viruses or some eukaryote.
Other already living things.
Yeah, exactly. We can't run these natural experiments today, because even if you do happen to find organic molecules produced naturally, like the Murchison meteorite, which contains amino acids, or if you go down to these hydrothermal vents, whatever molecules that are produced are going to be eaten up immediately. And so you have to, but it's a goalpost shifting, isn't it? Because they say you can't do it, and even if you can, it doesn't matter. And so I tend to say to that, then why are we even having this conversation? If you're not going to accept any evidence, against your position, then it's just intellectual dishonesty.
Well, I think their argument is normally that it takes the scientists have to like put it in the oven, take it out of the oven. They have to like deliberately, manually manipulate the data. They can't just let it run on its own, and that I think is the main criticism.
Well, I mean, sure, but you also look at things like the original Miller-Urey experiment. They literally just threw these chemicals into a set of tubes, and they said, we're going to let this run for a week, right? There was just this hyper cycle of water and ammonia and hydrogen sulfide and all that, and they're heated up, they evaporate, they condensate, they're electrified, they does that all over. And so in a sense, they did just let it run, and they produced several amino acids, right? But of course, they're like, oh, well, it was still perfect quantities of whatever materials you're using, which I don't really understand what that means. How would you know what the perfect quantity is? If
yeah, I think the thing. Well, it's not. It's like it's perfectly filtered, so it's not. There's not like a bunch of corruptors in there, because if there were, well, then everything would fail.
Well, I mean, even researchers do actually look at different ways to introduce contaminants into their systems, which is what happened with ribose when they figured out that ribose on its own would condense into tar. But if you introduce borate into the system, well, the ribose can fold rather than becoming tar. So even with contaminants, these can actually help stabilize, your molecules.
It's just harder to predict because we don't know what the contaminants are going to do. So it's simpler to make it pure to see what the results are going to be more reliably, rather than introducing more factors, which makes the test more complicated, right? Something like that.
Yeah, exactly. And this is really why I focus on sort of the evolution part, because if you can get them to acknowledge, if you get them to the point where they're saying, okay, well, all right, fine, there was a last universal common ancestor, explain to me how life originated, I would say, I'm done here. If you want to believe God did it, you're on your own, fine. But if you accept universal common ancestry, you have removed yourself both from young earth creationism and intelligent design. So I'm like, I win, Okay.
Yeah, you mentioned the asteroid. Do you think that those are common throughout the universe, or do you think that was? I think one of the hypotheses was that was from Earth, and that an asteroid hit Earth, and that it shot it up into the solar system, and it just came back or something. Like, do you think those are pretty common throughout the universe?
Oh, they're absolutely common. Yeah, the Murchison meteorite. Yeah, it had amino acids on it. I can put that inside chat. Murchison meteorite. Yeah. Organic molecules are easy to produce. You have moons like Enceladus and Titan, which have methane, which is the simplest organic molecule, because it's a carbon with 4 C's, or 4 H's, sorry, 4 hydrogens. Organic molecules are the easy part, I would say. The hard part is actually getting life, because you can find organic molecules everywhere. It's not really too difficult to produce those.
What's the difference between an organic molecule and a non-organic molecule?
Organic molecules, at least by our standard of life, are carbon-based. They have carbon-hydrogen bonds, whereas inorganic stuff doesn't. So, like, the simplest organic molecule is methane, right? It's 1 carbon, 4 hydrogens, and then you can go up from there. You got ethane, which is 2 carbons and 6 hydrogens. And so you have to have this sort of carbon hydrogen base for organic life, at least according to our definition. Now, there could be silicon-based life somewhere out in the universe, that's a totally different story. But
yep, totally, they're going to be real, and they're going to come and just dissolve us like the Borg. We're going to be the Borg,
right?
So, what did you think about your debate with Fuzz Rana?
It was good. I do want to say, I apologize if I'm not answering any questions satisfactorily. I would recommend, someone's at the door. I would recommend someone who has a much better and deeper understanding of biochemistry and origin of life stuff than I do.
Or do you have
Uh,
any suggestions that I can email, contact?
you could try talking to like Dan Stern Cardinelli.
Could you put his name in the chat so I can?
Yes, absolutely. Dan Stern. I'm trying to think. I don't know a whole lot of biochem people. Maybe PZ Myers. You could talk to him.
He's hard to get a hold of unless you're on the more leftist end of the spectrum.
Oh, well, he, those are a couple of guys. Those, I feel like I'm, forgetting someone obvious, and they're going to be like, "Hey, why didn't you call me?" And whoever it is, I apologize, but I don't really know a whole lot of biochem people who are on like our side of this issue. I know
Dimitar Kunev. Do you know who that is?
Who?
Dimitar Kunev. Scott Duke asked about him. Dimiter Kunev. I'll check him out too.
Yeah, sure. There are lots of biochemists. I know one guy, but he doesn't really do this sort of stuff. And so, but yeah, unfortunately, I don't know a whole lot of biochem and cell biology people. But Matthew Herron would probably be another good person to talk to. He's more of a cell person. Matthew Herron. My debate with Rana, with Dr. Rana, it went well. He, it's cordial. We're very cordial with each other. He didn't really have a model, which I was expecting. I suppose in one sense, I was praying for this, and I was a little nervous because
you were praying for this.
No, I was preparing for this.
Oh, gotcha. I was like, praying to who?
To Vishnu. But I was preparing for this, and I was a little nervous because he's a smart guy, and then when we sort of got there, I gave my opening statement, and he gave his, and I was kind of like, I didn't really hear an argument in there, and so we just kind of went from there, and he didn't really have a model, which I did expect. Intelligent design people really don't have a model of biodiversity. Creationists sort of have to. They
dogs come from dogs.
Exactly right. Yeah, you have the whole kind thing, but of course they don't work this out to any rigorous extent. As soon as you start sort of probing them on, well, what's the limit of being a dog? And then you go to the fossil record, and suddenly they shut down.
Is a Chihuahua a dog? Is a bear a dog?
Is a bear a dog? Yes. Yeah. The earliest bears were, the earliest dogs were plantigrade rather than digitigrade, like all modern dogs are. So were they dogs also? And then you kind of have to ask, well, does God create successive transitional groups to fool us?
Yeah. I love the examples. Like there's that one, the musk deer, the deer with the fangs on it. Like, so which one is this? Is this a deer? Is this a wolf? What is this?
Oh yeah. 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 It's funny there. I don't think there are any intelligent design people or young earth creationist people who are active in the debate circuit who are organismal people. Which, if you're debating universal common ancestry, you kind of have to be.
What do you mean by organismal? You mean the diversity of the organisms or something?
Yeah, you have to be, I think, fairly well acquainted with the diversity of plants and animals. But they're kind of like, oh, we got dogs, got cats, got cows, and that's about it. That's the whole thing.
Yep, there are only dogs, cats, and cows in creationism. You heard it here first from Jackson Wheat. That's correct.
Elephants are just large cows. What did you see that show? Is it Miracle Workers where Steve Buscemi is God?
Yeah, I think so. I think I saw that a while ago.
He's like what is a cow? It's like a big dog you can drink from. And what is a dog? A little cow you can be friends with.
So you mentioned that Fuzz didn't have a model. What do you think a model from a old Earth creationist would kind of look like? What would it mean for them to have a model?
Well, I don't think functionally it would be that different from the intelligent design viewpoint, but neither of them. Well, I'll say this: I think a model for them would be there should theoretically be at least one event in the history of life which was designed. And I know most of them love to jump to abiogenesis, and I say, well, I'm not
The
an
Cambrian
expert in that.
explosion is another one.
Yeah, I preempted that response, which is I made several slides for that, um, because that view is largely derived from Steve Meyer's 2013 book Darwin's Doubt, which is terrible. It's so bad. In his when he wrote that book back in 2013, or when he published, sorry, um, he was wrong about both of his major arguments, and it didn't really matter. And then he published another book, I think in 2015, where he had a bunch of people from the Discovery Institute sort of defend his book against all the criticism. And it was wrong then. It was wrong to say there are no predecessors of Cambrian animals in the Precambrian. That was wrong in 2013. It's dead wrong today. The more we find, the better technology we get, so we can do better scanning and look at these fine details and things that we didn't have back in the 80s, like when Stephen Jay Gould was writing. This has really revolutionized how we look at fossils and how we understand the history of life. And so when they bring up things like the Cambrian, or when the Cambrian explosion, and they say, oh, it seems like they just came out of nowhere. No, they didn't. Here's a set of fossils from the Precambrian that are either in the crown group, so they're actually part of the phylum itself, or they're in the stem group, so they're transitional fossils. Things like Eulingia, which is a stem panarthropod, and stuff like that. And so I don't know of anyone who is looking at these stem fossils in the Ediacaran and then saying, oh, I guess we need to fix our arguments now. No, they're just kind of ignoring them. And so every time I have the conversation about the Cambrian with the creationist or intelligent, well, it's really just more the intelligent design people, because creationists are like, there was no Cambrian explosion. I say, have you heard of this fossil? No. Have you heard of this fossil? No. Well, okay. All right.
So would you say there was no explosion? It was just the exact same rate of transitional growth as normal, or do you think there was actually an increased rate of diversity during that time period?
Oh, yes. I would say there was. You have sort of a couple stem groups in the Ediacaran, and then you have this flowering out in the Cambrian. When you look at the trace fossil record, for instance, you have a few trace bilaterian fossils in the Ediacaran, and then you have increasingly more of them as you get into the early Cambrian and go from there. So yes, it's not just a taphonomic artifact as some biologists had proposed previously. There is radiation. It's not ridiculous to the extent, that like all the animal phyla were appearing in the Cambrian.
It came from two, an Adam and an Eve.
Yeah, exactly.
An Adam worm and an Eve worm, and they just all came from that. It's beautiful.
Yeah, exactly. Yeah, Richard Dawkins' book, The Ancestor's Tale, he discusses the Cambrian explosion in the Velvet Worm's Tale, and he gives a couple of quotes from some biologists who said some really wacky things about it. But yeah, I mean, there was a real event based in part on the bioturbation of cyanobacterial mats, and the fact that you have multi-tiered ecosystems now, as opposed to sort of this flat space where everyone's just kind of grazing on the mats. And so, yeah, but it sort of becomes me just talking at them about all these different fossils, because they're not paleontologists, and that's not their fault. It's just they need to do better research on this topic.
But then they wouldn't be able to defend God. So no. But that would be, I think, the argument that Stephen Myers makes now is that, well, yes, all of these have been discovered, but there was still an explosion. There was still an increase that is somehow inexplainable in some way. So what caused that increase that was not there before? Why was there this just random increase in molecular life during that time period?
So, well, I mean, he just ignores the fact that we do have early or Ediacaran fossils, because his whole argument was there's nothing. There's nothing. You got some weird things, and then you got life as we know it, which is no, we don't. But even still, there was sort of a series of events. You had, so in the Cryogenian period, from 720 to 635 million years ago, you have these ice ages, these worldwide glaciation events, which occurred as a result of Rodinia accumulating. And so Rodinia kind of broke up and sort of moved southward. And so it wasn't in the tropics anymore, so there wasn't the rate of erosion, which dragged
The
carbon
continent
dioxide.
of Rodinia, correct?
Yes, the supercontinent of Rodinia. So there was only one landmass to rule them all, and that was Rodinia. And so once it kind of moved out of the equatorial region, it couldn't be eroded at the rate that it was, meaning that there wouldn't be another glaciation event. So then following this, you get your first first large macroscopic life, and that's the Avalon, or sorry, that's the Atlantean Formation. And then there's another glaciation event, not as severe as the Cryogenian ones, but you get another glaciation event, and then you have the Avalon explosion. And so this is, animal life is proliferating in its early stages, sort of at this point. And so by the next assemblage, which is the White Sea assemblage, you have a diversity that has built up now of different organisms. You have early bilaterians, early mollusks, or probable early mollusks, things like that. And so then you have an extinction event at the end of that one, which leads to the Nama assemblage. And so you have reduced forms of the weird Ediacarans with respect to the animals. And then you have an increase in oxygen. And so this increase in oxygen helps sort of kick off the Cambrian explosion. And now you have, with more oxygen, you can get bigger, because we do cellular respiration. So our oxygen is used to fuel the creation of ATP. So you have more cell energy, so you can get larger. And this means you could have multi-tiered ecosystems now. And so the evolutionary arms races that occurred between different organisms took off from there, which is what helped generate the diversity of life that we do see in the Cambrian.
So kind of like a punctuated equilibrium kind of event.
Yeah, basically, yes. You have these different events which are causing bursts of diversity. Then some of them get wiped out. Then you have another burst of diversity. Some of them get wiped out, and that's kind of what you see throughout the history of the Phanerozoic also. So from the Cambrian to today.
Do you have a position on Stephen Jay Gould's punctuated equilibrium as opposed to the more Darwinian natural selection kind of approach?
Well, they both utilize natural selection. The debate was more between, is life always gradually evolving, which is the phyletic gradualism approach, or are there periods of stasis followed by periods of punctuation where you have this big burst of diversity? And well, it's actually both. There are lineages you can look at where you see gradual evolutionary change over long time periods, or if you look at things that have really, really good fossil records like phytoplankton, forams, diatoms, things like that, you can see these gradual evolutionary transitions which are just magnificent, which also completely refute the global flood. But because how could you have them tiered like this? But punctuated equilibrium also occurs when you have an extinction event. You have a wipeout, and then you have a rebound of organisms, and then they fill the niches, and they sort of become static in their morphology by and large, and then they get wiped out. Then you have a refilling of the niches, and so then they become static. So it's both, depending on which organism you're looking at.
Do you think that it's more the gradual than the punctuated equilibrium, and that just happens at increments rather spread out usually?
Well, in a sort of sense, in one sense, you sort of have constant, or you have gradualism in every generation, because you have genetic drift, which occurs in every generation. So you change the frequency of alleles. And so there's a sort of gradualism that occurs in every generation. So I guess in one sense you could say, sure, gradualism occurs more. I mean, the genes are changing regardless, but the outward morphology may be relatively static. Like crocodile, or the crocodilian form, I guess you might say, goes way back to like the Triassic. But there was a diversity of crocodilians. They lived in different environments. Some of them became totally marine, some totally terrestrial, some, ambush predators. So though they outwardly look crocodilian-like, they were still evolving.
And what about the argument that, Darwinian evolution is wrong because of evolution 3.0, Evo, Devo, and all those other things?
The extended evolutionary synthesis crowd, they're kind of wacky. They seem sort of removed from like mainstream biology because they say things like, because they say that if you ask someone like PZ Myers, has Evo Devo been integrated into the modern synthesis, he would say, well, yes. But if you go and ask James Noble, I think is his name, one of the EES guys, he's like, we need to integrate Evo Devo and epigenetics into biology. And all the other biologists like, we have been. What are you talking about? And they're kind of wacky that they say things like that. I mean, these are just normal parts of the evolutionary theory already, and they have been for, in the case of Evo Devo, since the 80s.
Yeah, awesome. I totally agree. That's the one thing is that, especially when debating the Muslim apologists, they say, Darwinian evolution is false because of there's these additions because it didn't get everything right in the first incarnation when Darwin wrote his book. Darwin is wrong.
Really? They say that?
Yes, they do. That's
wild.
Quite hilarious. Are there any creationists who you would like to debate, who you think do produce a good argument? Like, would you want to debate Stephen Myers or Dembski or the other guy, I forgot his name?
Oh, yes. I would love to have a conversation with Meyer over the Cambrian, because I know he can't defend it. But it's kind of amazing. He writes like a 500-page book, and then it's just terrible. Yeah, but I mean, I doubt any of the big people would ever, I'd ever get the chance to talk with them. Funny enough, in our back and forths with Long Story Short on my channel, they actually wrote an article which mentioned us at Evolution News, which was written by David Klinghoffer. So I guess I'm on their radar technically, but I don't know. They have more interesting intelligent design people tend to have more interesting arguments. Creationists tend to be just flat wrong about everything, whereas intelligent design people are kind of wrong with a twist. They like to very selectively use their facts, but in such a way that it sounds like they're right, but they're not.
Yeah, I'd like to debate James Tour. He's got probably the most convincing argument for most people about the origin of life kind of a thing. So he's one I'd like to chat with.
Oh yeah. Oh, which actually reminds me, John Perry from Long Story Short. John Perry. He would be a great person to discuss origin of life stuff with.
Nice, definitely. I'll contact him for sure.
Yeah, Tour is interesting. He's an interesting character in that here you have a chemist who is very vocal about, nothing has changed in origin of life research for, like, what does he say, 50 years, something like that.
We've made no progress.
Which just factually isn't the case. The concept of where the origin of life occurred is radically different today than it was 50 years ago. We have done lots of experiments with how molecules interact, and there are side benefits to doing these sorts of experiments, helping us to figure out how certain molecules interact can also impact medicine, right? If these things act this way, well, could we use that in a human system to deal with some type of disease, or with shuttling certain molecules across a membrane barrier, for certain hormones that we have to give to people.
Yeah, I remember there was
So
a paper about that, about using gold or something as a marker to I think track cancer or something.
yeah, absolutely, yeah, sure, yeah, that kind of stuff. It's ludicrous to say that no progress has been made. Do we know everything? No. Will we come to a point where we maybe have a working model of abiogenesis? Probably at some point. I don't know when, but it's ridiculous to say that like we don't know anything. Uh, I don't know how often you've had the abiogenesis conversations with creationists and ideas, but it always seems to come down to a protein. Here's one specific protein they're really in love with, and they just assert over and over it couldn't have evolved because they just love this protein.
Yep, that's usually what they say. They say this really complicated molecule. You can't generate this really complicated molecule in a lab, therefore life is impossible.
Yeah, exactly. And yeah, it's something like I remember one of them was talking to me about the topoisomerase, and I looked it up. Turns out topoisomerase probably evolved by like a couple mutations from this other enzyme. And I was like, well, that deflated your whole argument.
Yeah, that seems to be the main stopping block for them, is that they want the full, most evolved form of life, whereas the origin of life was nothing like what life looks like today. It was far simpler, and so trying to compare the life as it is today to life as it was when it originated is not a realistic comparison, because we don't even have like a full definition of what life really is, because we don't know how to define the origin of life, and we could have like already had what would be necessary to produce life we have today if given the right environment. We just wouldn't know it yet because we don't have the information to accurately define what the original life form would have looked like.
Yes, absolutely. Yeah. Yeah, that's a huge debate in biochemistry is, well, when is a protocell, which is doing metabolism, a cell? When do we consider it an independent living system? Because whatever criteria you come up with, there's some organism who doesn't do it, or there's some inorganic matter that does do it, right? Viruses are the best example. Here's something which isn't alive by most people's standards, but it replicates, it mutates, it evolves, it uses your metabolism to do its functions, but it's not alive, right? Or look at some bacteria like Rickettsia or Chlamydia, which can only live inside of cells. They can't be cultured on their own outside of cells. They have to live inside cells. So are they alive? They're bacteria. And or heck, prions. Prions are just proteins. It's just a protein that reproduces, right? It's wild.
Yeah,
And
that's
yeah,
absolutely one of it's like the same just species problem where we try to define, well, when does one species turn into another species? The same kind of a thing. We don't have any way to prove one way or the other that it's really a species, because it's kind of just arbitrary. This is they're different, so we're going to call them different.
yeah, exactly. Yeah, these are useful constructs. Although, well, species or alive, these can be useful constructs, but even biologists apply a sort of essentialism in which it has to be this or it isn't. And it's kind of funny because the whole thing about like evolution is that's just wrong. There is no essence of leopardness or of rabbitness. There are animals who look increasingly or decreasingly like the modern leopard or the modern rabbit. And you can take both of these and go back to a common ancestor who was not a leopard or a rabbit. And so when you try to say, or when not you in particular, when biologists try to say, how do we define a species? Well, we can sort of define it. We can give it a rough estimate, but trying to say this is a species or this isn't is kind of pointless. It can also have ramifications for conservation, right? If some certain group is considered to be the same species, well then it's not going to receive a certain protection. But if it's considered a different species, well now the government can look at it and say, oh well, that's an endangered species, so we're going to provide them with some protection. And the same with life. There are things that have varying degrees of lifeness. Life just isn't. It's not a. What is it? There was that old idea that life was like a chemical, and it was disproved with the
Oh,
urea experiment.
yeah, I do remember that.
Vitalism.
Yep, vitalism.
Yeah. So yeah, it's like that. There's no life essence, no life force that we can touch, um, and so it's just a it's a it's just another gradient, I guess you
Definitely.
could say.
We got a super chat, by the way, from Marty Mad Scientist. He asked, is the theory of biochemistry that is most efficient way of breaking down complex chemistry using energy from radiant legit, i.e., is life inevitable?
That also that is an interesting question. Life does have to use biochemical gradients, and uh, when we're for instance, cellular respiration occurs along the proton gradient in the inner membrane of mitochondria. So you're shuttling electrons across this protein complex, which is then pumping um, protons uh, back into the mitochondria so that you can make ATP. And it's funny because these gradients are also or hydrogen sulfide gradients are also uh, in existence at alkaline hydrothermal vents. Woo! How curious, you know?
Oh, and what about, have you heard about the studies that quantum effects on DNA?
No.
Because I've been seeing that more recently in a few papers of how the quantum effects we've discovered affect DNA and somehow. I'm a little skeptical, but it's an interesting topic.
I
Uh,
don't know.
we got another question. What is the most interesting argument you've ever faced from Hertz?
What is the most interesting argument against evolution, or
I assume so. Yeah, against evolution from the ID people, or any god in general, kind of a thing.
um? Well, I don't know. I think the Cameron explosion was kind of an interesting one. That's one that I've done compared to other arguments. A lot of research. I've read a lot of papers. I've read even uh, books on like the Cameron explosion, and I think armed with that knowledge, you can show pretty easily that yeah, no, the ID conception of it is totally garbage. Uh, or I like having the baraminology argument with creationists. Where do you draw the line at kinds? Because they can't. And if you look at the Faisal Rana debate I had recently, um, it's the same for intelligent design. They don't have to have a baraminology aspect to them. Nothing about intelligent design necessitates having created kinds or types, whatever, but they do it to themselves because they don't like the idea of things being complex but coming from a natural source. So they kind of do it to themselves.
Yeah, they make the claim, and so they have to try and back it up, even though it's a ridiculous claim.
Yes, exactly. Yeah, and you can see that happening over and over in my debate. It's kind of like why this but not this? Well, I'll have to think about that, which is fine. That's a perfectly valid answer. But if you're saying intelligent design is definitely true, but then you don't really have a model of anything in it, you know.
What do you think are the best examples of transitional fossils?
Uh, well, I think TikTok, is one of the best. Still, but part of the reason I like it is my favorite transitional fossils that I always go to: Microraptor, Diarthrognathes, Tiktaalik, Strika myrma, which is a little wasp ant. They're all predicted. They were all specifically predicted under evolution. Nothing else. If evolution is true, or Archaeopteryx, that's another one, then these should exist in the fossil record. We should have in the fossil record a skeleton that has characteristics of two different clades, which on the other view are unrelated. This is inexplicable on the other view, which is creationism.
Yeah, I totally agree. I think that was one of the best evidences for evolution that we predicted these things before we discovered them. And I think for Tiktaalik, they predicted it in to a 1 kilometer range or something. They said we're going to find it in this location in this geological sphere, and we got it right. That's incredibly impressive.
Yeah, and Schumann et al. yeah, they went to that one island way out in the Canadian Arctic, and they were like, well, this is where the strata occurs, so this is where we're likely to find this thing. And Microraptor came from this guy named William Beebe, and he said he looked at birds. He was an ornithologist, and he looked at birds. And he said, well, birds in their young stages have these little flight feathers which develop on their hind legs, which then don't function in flight during their lives. But the ancestor of birds should have had flight feathers on their front and back legs, or front and back limbs. And there you have Microraptor, 2003. So like 100 years later, they finally found it. Darwin predicted Archaeopteryx in saying that the ancestor of birds probably had unfused wing fingers. Archaeopteryx does. Diarthrognathes was one of the mammal-like reptiles, the synapsids. And Robert Broom said, if mammals are in fact evolved from reptiles, then they should have a very particular jaw joint in which you have two types of joints going on at the same time, because one set of bones is moving up to become the ear, or the inner ear bones, while the other bone is enlarging. And so, yes, some specific characters that must have existed in this fossil if this thing, if we did in fact have this ancestor in our line. And look, there it is.
Well, that actually brought up another question when you mentioned Archaeopteryx and the unfused fingers. The birds are dinosaurs, but they're not T-Rex. So how are birds dinosaurs, but they're not directly descended from T-Rex?
So you have the three major groups of dinosaurs. You have ornithischians, so the bird hip dinosaurs, which are not related to birds, or are not closely related to birds. Things like Stegosaurus, Ankylosaurus, Iguanodon, that's the ornithischians. And you have the sauropods, the long neck dinosaurs. Then you have the theropods, so the meat-eating dinosaurs. And the earliest meat-eating dinosaurs the earliest theropods have the lizard hip. They're called the Saurischian hip, because the pubis points forward rather than backwards, as in birds. Well, as you kind of go along through the evolutionary lineage of theropods, it starts kind of shifting backwards to the bird position. And so you have this group called Coelurosauria, which is tyrannosaurs and birds and dromaeosaurs. So tyrannosaurs are in that group, but they're kind of but they are more distantly related to birds than dromaeosaurs are. So like Velociraptor, Deinonychus. And then we have a smaller grouping, which is called Maniraptora, which includes dromaeosaurs, birds, and a couple other groups like Therizinosaurs, which also have feathers. And then you have paraves, which are like just the birds and their very closest relatives, like dromaeosaurs. And so they're all close together, but they're in these separate little nested clades. And so birds and dromaeosaurs are the closest relatives, because they both share feathers. They both have this backwards pointing pubis and all these sorts of features in common, such that some have sort of flipped based on paleontological discoveries. The researchers are like, well, is it an early bird? Is it a dromaeosaur? You start getting to the point where the lines are really super blurred. Things like Anchiornis.
So the ancestors for our common birds were they were also smaller dinosaurs, like the same size as birds, not bigger dinosaurs like T-Rexes, correct?
Right. Yeah, they would have been small. We're talking Archaeopteryx is very little. You see the big picture from the Solnophan, the famous one, but they're little itty bitty guys. They're like crow sized. And so yeah, they're very small.
Have there been a lot of instances in that where what we thought about the relation between species ended up being wrong, kind of like how we thought that elephants were more related. I think it was elephants were more related, or hippos were more related to elephants, than we discovered they're actually more related to like whales.
Oh yeah. As genetics has gone along. Well, you have to remember the first phylogenetic trees, which were made way back in like the late 1800s by Huxley and others, because Darwin was like, okay, here's my idea. Organisms are related in this fashion. And so Huxley, Haeckel, and a couple other guys were like, okay, So we're going to take sort of the data that we have and try to figure out who's related to who. And these trees were based totally on morphology. So how does it outwardly look? And funny enough, whales are actually considered to be in the artiodactyl group, the hoof mammals, the cloven hoof mammals, way back in the 1800s. And they did this based on like the way that I think it was the similarities in the skin or something like that. It was something weird. Is like, really, that's how you determined it? Okay, but they were right, you know. But for a lot of things, they were way off. Like brachiopods and mollusks, for instance, are two different groups. They're not, you know, in the same exact group. There are lots of different kinds of worms, like flatworms, which are actually, there's actually a couple of different groups of flatworms, which are not closely related. And so genetics kind of torpedoed a lot of the morphological groupings. But now that we have these genetic groupings, we have really the definitive relationships among them. Right? Yeah, humans are definitively the closest relatives of chimps, things like that. Even though that was kind of already known.
JB's making the creationist argument. Well, we can't trust the isotopes from the like testing of the ages of the rocks. How can you be right about those?
Yeah, you can. It's well, again, I'm not a geology person either, but yeah, you can. There are, one of the great things about radiometric dating is you can test multiple, if you use the correct dating method, you can test multiple dating methods on the same rock and get very similar results. But you can also do this stratigraphically. You can look at the fossils. There are these things called index fossils, which are contained within strata, and you can look and you can correlate these across the planet. So a strata of one age is going to contain a particular set of index fossils, but it's not. But the layer above it and the layer below it are not going to contain these. And you can do that for like every layer. If you just go to Wikipedia and look up like each epoch, and it will tell you which foraminifera it's correlated with. And so you can first do that. And so then you can also use radiometric dating methods, and you're going to get this increasing age as you go further back, which, if it were young Earth creationists, if the young Earth creationists were right, shouldn't we expect everything to come up with like 10,000 years or 4,000 years if it's, you know, deposited by the flood?
Well, I mean, you got the density of the bones is different, so they just settle on different layers. Come on.
Oh my goodness. It depends on which. Well, I mean, if that were the case, you know, we should expect like sauropods to all nest together, but
we can't see the bones from the past because they've all turned to rocks, so they could have all been different.
Goodness.
Like, come on.
Oh, so many rescue devices there.
Well, that's one of the things in philosophy, the problem of underdetermination. You can always add in ad hoc reasons to explain literally everything. So there's never a way to truly falsify any theory. It's only falsified if it's a very specific prediction. And
sure, yeah.
You're right. That's the rescue device that everyone goes for in human psychology. I can make up an ad hoc reason to make that work, and we think that that's actually a justification when it's really not. One of the human fallacies, biases, delusions, illusions, all of those things, which, as Darwin said, must have had some kind of evolutionary benefit, which is why we have these silly beliefs.
Yeah, I mean, and the thing is, a lot of times when you come up with, or when not you in particular, when someone comes up with these ad hoc reasons, a lot of times they are actually testable. They provide testable predictions when they say things like, well, the bone density is different. Okay, well, then we should expect the heaviest ones at the bottom and the lighter ones on top. This would make testable predictions that we should be able to either confirm or falsify. And they keep coming up with reasons as to, no, we can't actually falsify or confirm this. No, we can't do that. Whereas, and that's what creationists do. But in science, you have to confirm or falsify this. If you come up with a model, you have to test it because you proposed it. And if you can't test it, no one's going to take you seriously. Right?
Definitely.
Only in creationism can you just make up models willy-nilly and just kind of assert it.
All right. One last question before we go. It was about one of the standing for truth. He always brings up this argument of the genetic decay. I forget what it's called.
Genetic entropy.
Yes. Yeah. What is your response to genetic entropy?
I, as it happens, did an entire video. Well, my team and I, we did a whole video on genetic entropy, and it's garbage. It is so garbage. Dan Cardinale regularly interacts with them on this topic, and when the guy who came up with, well, the guy who came up with genetic entropy, John Sanford, well, he didn't really come up with it. He just sort of repackaged an old argument that was already in circulating in creationism for the past like 70 years. But he put a nice little new gloss of paint on it. And
so he's the Elon Musk of the Hyperloop, essentially.
basically, yeah. He claims that there's this degradation in this mutational degradation in all organisms because things can't ever go uphill; they can only go downhill. And so, he claims deleterious mutations will accrue indefinitely. They can never be sort of overshadowed by beneficial mutations, which is wrong. It's based on an incorrect reading of a 1974 paper by Moto Kimura. And so Sanford misrepresents this paper. This single misrepresentation is the core of his argument. And once you show that he's wrong on it, the entire argument is dead. It's just dead from there. It's already over. And so, because he claims Kimura made this graph in which Kimura excludes beneficial mutations because if you look at this, the population genetics, the mathematics of it, beneficial mutations hugely outweigh harmful mutations. And so when they arise, they're going to be more rare, obviously, but they're going to cause like selective sweeps where a whole bunch of harmful mutations you had previously are all just kind of wiped away. The effect does not in any way outcompete the beneficial mutations. They are way more powerful, and this has been confirmed empirically in the field. This is mathematically true. Genetic entropy, on the other hand, even Sanford admits there are no empirical studies which demonstrate genetic entropy. There are 0 lab studies. The bacteria experiments, the long-term fly experiments, none of them have degraded out of existence. Again, he uses an example of H1N1. He says, it died off, but he asserts that the pathogenicity of H1N1 equates with the fitness of it, which is just wrong also, because if you know anything about virology, being less pathogenic means you are more likely to spread. And so again, another core tenet, which is already dead, but standing for truth and his brain trust, they don't care. They don't read the literature. They don't know anything about basic biology. And so,
yeah, I think they represent as just the intuitive argument that if there's a ton of mutations and the rarest kind is the beneficial kind and the detrimental kind are more common than the beneficial kind, even if you get a beneficial kind, then along with that is going to come some kind of negative mutations in addition to that go along and are transmitted to all the offspring from whatever the beneficial mutation is. Therefore, for every beneficial mutation, you'll have like 10 detrimental mutations, and therefore the detrimental mutations will build up just intuitively.
Right. Yeah, exactly. That's what you would think, right? You would think, or you could even say like 100. For every 100, detrimental mutations, there's going to be one little lone beneficial one, but it's just 1 against 100. It doesn't matter. Well, actually, yes, it does, because this is how natural selection works. Natural selection is going to positively select for that one beneficial mutation. And so it's inherently going to be way more powerful than all of your slightly deleterious mutations that you've accumulated. And so it'd be more like the effects are more like 100 to 1,000 in terms of the beneficial mutation, rather than it's just they're all on the same playing field. It's just a matter of 1 to 100.
Oh, to clarify what I mean is like in the same cell, in the same animal that has the beneficial mutation, there's also going to be detrimental mutations. And so for any beneficial mutation, there's also going to be detrimental that also happen in that species. So it doesn't matter how many beneficial mutations you get, you're always going to have more detrimental ones in addition in the cell that you can't get rid of, because the same for any mutation pattern, there's always going to be for one beneficial, a whole bunch of negative ones. Right. So even if all the other ones get taken out from the species, the ones that happened in correlation with the positive one, you can't get rid of those.
Right. Yeah. But I mean, again, even though these will accumulate, these slightly deleterious ones will accumulate, they're still way outmatched in terms of what they do to the organism or the population, the species, in comparison to the beneficial ones. And Sanford and guys like Basner, who also write papers with him, they just ignore this fact. This is just a basic fact of population genetics, and they just kind of ignore this. They're like, eh. They basically try to place like all mutations on the same sort of effectiveness. Like one beneficial mutation and one harmful mutation will like cancel each other out. It's like, no, that's not how this works at all. And so all the arguments for genetic entry are bad. There's no empirical evidence for it whatsoever, no mathematical evidence for it. They're just potato bad.
Awesome.
And that's why no geneticists take it seriously.
It's trash, garbage, done.
Exactly. Yeah.
So do you want to talk about a little bit of what's coming up on your channel? Anything planned? Anything you want to talk about before we head out?
Well, let's see. Upcoming different videos. My hope for some collabs and things like that. I'm always trying to come up with new ideas for videos. I was kind of slow recently because of school stuff. RJ and I are working on the Rocks Were There Volume 2, so there will be 2 volumes of that, and it should be will be the second volume of the most comprehensive creationism refuting books out there. That's really about it. That's really all I got.
Cool. Well, thanks for coming on. I really appreciate it. It was a fun conversation. I'd love to talk with you again sometime, and
Absolutely.
love to see you on Modern Day Debate, crushing some more creationists occasionally.
Yeah.
Awesome.
Yeah, absolutely.
Thanks again for coming on. I really appreciate it. Have a nice day.
Thank you for having me. Yeah.
See you later.